BSc Zoology, 3rd semester, University of Mysore, Session 32, Gametogenesis, in Gametogenesis, Spermatogenesis. Unit 2, Developmental Biology, Gametogenesis, Spermatogenesis. Here the students, the objectives of learning is to learn about gametogenesis, to learn about spermatogenesis, and to learn about the formation of spermatids, and also to learn about spermiogenesis. The session outcomes. Here the students enabled to learn about the definition of gametogenesis. The student learn about spermatogenesis. They learn about the formation of the spermatids, and they learn about the meaning of spermiogenesis. Coming to the definition of gametogenesis, in embryology, the process by which the gametes or the germ cells are produced in an organism is called as gametogenesis. There are two types of gametogenesis. One is oogenesis, another one is spermatogenesis. The major difference between the oogenesis and spermatogenesis is the formation of the egg cells or the ova is called as oogenesis, and the formation of the sperm cells or the spermatozoa is called as spermatogenesis. This is the basic difference between the oogenesis and spermatogenesis. Coming to the definition of the spermatogenesis, it is the origin and the development of the sperm cells within the male reproductive organ, the testis. The sperm cells are produced within the testes in the structure called as seminiferous tubules, and once the sperms are matured, it is transported through the long seminiferous tubules and stored in the epididymis of the testes until it is ready to leave the male body. Here this is a structure showing the male reproductive organ, that is the testis, and the process of spermatogenesis, how the spermatogenesis occur through the primary germs, primordial germ cells, then primary spermatocytes. Through the mitotic division, the spermatogonium is converted into primary spermatocytes, and through meiotic division it is converted into secondary spermatocytes, and through meiotic division 2 it is converted into spermatids, and through the process of spermiogenesis the spermatid is converted into the mature sperms. This is the actual process involved in the spermatogenesis. And coming to the spermatogenesis, here the mammalian spermatogenesis is highly synchronized, regular and long and extremely complex processes of cellular differentiation by which spermatogonial stem cells is gradually transformed into highly differentiated haploid cells, the spermatozoon. The differentiation involves three distinct classes of germinal cells. One is the spermatogonia, the spermatocytes, and the spermatids, which is usually they are arranged in the concentric layers in the seminiferous tubules. In adult mammals, spermatogenesis is a continuous process which can be divided into two distinct phases, and it is characterized by specific morphological and biochemical changes of nuclear and cytoplasmic components. The two phases includes the formation of the spermatids, that is through the process of mitosis and meiosis, and the spermiogenesis. The formation of the spermatogenesis is further divided into three phases. One is the multiplication phase, and this is also known as proliferation phase, and it is a renewal of spermatogonia. So during this phase, the diploid spermatogonia, which is situated at the periphery of the seminiferous tubules, multiply mitotically to form spermatocytes and also to give rise to new spermatogonia, stem cells, and enter into the phase of growth. The next phase of growth, and you can see here what I have shown before in the slide, that is spermatogonia, the primary spermatocytes, the secondary spermatocytes, and the spermatids, and developing into matured sperms. And the next phase of development is the growth phase, and during this phase the limited growth of spermatogonia takes place and their volume becomes doubled, and they are now called as primary spermatocytes, which are still diploid in number And now this primary spermatocytes enter into the next phase, namely the maturation phase. So here the maturation phase, the primary spermatocytes enter into the prophase of the mitotic or maturation division, and meiotic prophase is a very complex process characterized by an ordered series of chromosomal rearrangements which are accomplished by the molecular changes. And during meiosis, first nuclear DNA duplicates and each homologous chromosome starts pairing, that is called as a synapsis, pairing of the homologous chromosomes, and longitudinally splits up into two chromatids, and both of which remain joined by the common centromere. And by chiasma formation, mutual exchange of some chromosomes material between the two non-sister chromatids of each homologous pair occur, and to provide almost indefinite variety of combinations of paternal and maternal genes in a gamete. And two chromosomes of each homologous pair, that is called as a tetrad, migrate towards the opposite poles of the primary spermatocytes, and now each pole of primary spermatocytes has an haploid set of chromosomes, and each set of chromosomes is surrounded by the nuclear membrane developed from the endoplasmic reticulum, and the first meiotic division as rule is followed by the division of the cytoplasm, that is what is called as cytokinesis, and which divides the primary spermatocytes into two haploid secondary spermatocytes. And these secondary spermatocytes, they undergo second meiotic or maturation division, which is a simple mitosis and produces the four haploid spermatids, and these are non-functional male gametes, and they become functional spermatozoa, and they undergo a complex process of cytological and chemical transformation, a process usually referred as spermiogenesis. This is also one diagram showing how through mitosis it is going to develop into the primary spermatocytes through the process of growth phase, and then primary spermatocytes developed into secondary, and then spermatids, and then spermatozoa. Next is spermiogenesis. It is important because the changes in the spermatids leading to the formation of the spermatozoa, it constitutes the process of spermiogenesis. So because of spermatozoa is very active and mobile cells, in order to provide a real mobility to it, so all the superfluous materials of the developing spermatozoa are to be discarded, and a high degree of specialization takes place in the sperm cells through the number of steps. So you can see here the number of steps takes place in order to change into the matured spermatids. So by the process of formation of the head, the changes in the nucleus, the Golgi complex, the cap phase, and the acrosome phase results in the formation of the tail of the spermatozoa. So this is about the gametogenesis, that is especially the spermatogenesis. Coming to the multiple choice question, the process of formation of the haploid male and female gametes is known as the following. One is implantation, gametogenesis, spermatogenesis, and oogenesis. You know that it is a gametogenesis. The primary sex organs in female is spermatogonia, the oogonia, the testes, and the ovary. So ovary is the primary sex organ in female. Diploid primary cells is in male is spermatogonia, oogonia, testes, ovary. It is spermatogonia. The process of formation of haploid male gametes is known as which of the following? Implantation, gametogenesis, spermatogenesis, oogenesis. Male gamete, so it is spermatogenesis. Diploid primary germ cells in female is spermatogonia, oogonia, testes, and ovary. The answer is the B, oogonia. These are some of the references. You can go through these books for further information. Thank you.